Hey guys, so this is just a quick video to help you learn the vocabulary for chapters 1 and 2. The first word, polysaccharide. A polysaccharide is a type of carbohydrate. Poly means many. Saccharose means sugar. So it's a carbohydrate that's made of many sugars attached together. So it's a long chain of glucose usually. Abiogenesis. This is a word that literally means without life generation, or generation without life. Let's go with that. And this was the theory for a long time, that living things just arose magically or by divine intention out of non-living things, that maggots could grow from meat or rats could grow from a garbage pile, and that babies just got planted there somehow. So it was kind of a silly idea when we look at it now, but that's what people thought, that living things came from non-living things. It was also known as spontaneous generation. All right, Peptidoglycan. This is an important molecule in microbiology. It is a molecule that's on bacterial cells, and it makes up their cell wall. So it's the peptido part is peptide is protein, and glyco meaning a carbohydrate. So it's both part sugar, part protein. And it's a structure that's important for building bacterial cell walls, peptidoglycan, and is not found in human cells. We don't make peptidoglycan. Ubiquitous. This is really not a microbiology word so much as it is like an SAT word. It's a word, big word that just means everywhere. It's all over the place. So germs are ubiquitous. Racism is ubiquitous. Evolution is the next word. Evolution is a scientific theory, and remember, scientific theories are well-evidenced, well-researched things that have a lot of evidence, not just an idea. So evolution is the process of organisms changing and adapting over generations through genetic changes. Cellulose. Cellulose is an important polysaccharide. It's a long-chain carbohydrate, and it makes up plant cell walls, so it's important for cell walls. And it's different from peptidoglycan. Peptidoglycan is important for bacterial cell walls. Cellulose is important for plant cell walls. So different types of cells have similar structures, but they're made of different materials. Pathogen literally means an agent of disease that causes disease. And so there's a lot of different bacteria and viruses and parasites. There's different microorganisms out there, but a lot of them are good for us or sort of neutral, have no effect on us. It's the ones that cause disease that we refer to as pathogens. Bioremediation is the ability of microbes to clean up the environment, to remediate biological, environmental problems. So there are bacteria that can eat oil or gasoline, and they can be used to clean up oil spills, or ones that eat plastic. They're trying to engineer ones that eat plastic to try to solve the plastic waste crisis. Biotechnology, so bioremediation would be a segment of biotechnology. It's just using biological organisms in a technological way. So the other aspect of biotechnology is recombinant DNA technology. So in some parts of biotechnology, we can just sort of breed bacteria or fungi to do the things we want them to do. But with recombinant DNA technology, we're literally recombining DNA, mixing DNA from different organisms, and putting it in a bacteria so that it has traits that we want it to have. So that's recombinant DNA technology. Macromolecules. Carbohydrates, proteins, and fats are the three types of macromolecules. Literally means large molecules. And those molecules are large, chemically speaking, but they also are known as macronutrients because we need them in large amounts. The other macromolecule that's not a nutrient is DNA, another large molecule. And they're sometimes referred to, those four macromolecules are also sometimes referred to as biomolecules because they're really important in biological organisms. Lipopolysaccharide, often just referred to as LPS, is a lipid, lipo, lipid. Polysaccharide, long-chain carbohydrate. So it's part lipid, part carbohydrate, and it sticks off of bacterial cells, specifically gram-negative bacterial cells, and it happens to induce a pretty intense immune response that leads to fever and other things. So it's something that's recognized by our immune response as indicative of a gram-negative bacterial cell. It's just something that sticks off of them on their surface. Triglycerides. Triglycerides are fats. They're a type of fat or lipid, and these are the ones that, like in your fat cells and your adipose tissue, you're storing triglycerides. When you eat food that has fat, saturated or unsaturated fatty acids, those are part of triglycerides. So 95% of the fat in your diet is from triglycerides. We use these for energy primarily, but we also use them to synthesize some hormones. Primary protein structure. So proteins have different levels of structure: primary, secondary, tertiary, and quaternary. 1, 2, 3, 4. The primary protein structure is simply the sequence of amino acids. Proteins are made of amino acids, and the sequence or order of them, the code of it, I suppose, is the primary structure. The secondary structure we'll talk about in class, but the other level of structure that's really important to understand is the tertiary protein structure. And this is the three-dimensionally folded, fully folded protein structure. So imagine a long string of beads, those being the amino acids, and then you sort of tie it into a big knot. That's the tertiary structure. And tertiary structure is really important for proteins to function properly. One of the major roles of proteins is that they act as enzymes, and enzymes are biological catalysts. They are just proteins that speed up chemical reactions. They bind to substances that are reactants in a chemical reaction, and they just speed up that reaction. And we rely on them in every cell. Antibodies are another another form that proteins can take. Antibodies are made of protein, and they are an important feature of the immune system. They're produced by B cells. Another macromolecule besides carbs, fats, and proteins is nucleic acids. So the two nucleic acids are DNA and RNA. So DNA stands for deoxyribonucleic acid, and it is stored in the nucleus of the cell. It's the genetics. It codes for all the proteins in our body. RNA, very, very similar stuff, stands for ribonucleic acid, and it is made in the nucleus, but then it goes out into the cytoplasm. It's sort of, actually, there's lots of different types of RNAs that we'll get into, but they're both nucleic acids. That's important to know. ATP is actually a single nucleotide of RNA, and so we can use it to build RNA, but it is also an important molecule in the cell because we use it for energy. It stands for adenosine triphosphate, and it has 3 phosphates, triphosphate, and when that last phosphate gets broken off, it releases a ton of energy that can then be repurposed to do things in the cell. So ATP is often referred to as the cellular energy currency. Taxonomy is just a field of biology that's involved in classifying things, living things, into different groups. And there's different levels of taxonomy, but we're not going to focus too much on that. There's 3 different types of cells, but the 2 main types that we are going to be talking about are eukaryotes and prokaryotes. So a eukaryote means true nucleus or a good nucleus. Eukaryotes have a nucleus and also lots of organelles, which are just like tiny little cellular organs that have different purposes. Prokaryotes are very simple cells structurally. They are just a cell membrane with some jelly floating around and some proteins and some DNA. But there's no nucleus, there's no membrane-bound compartments. Prokaryote means before the nucleus. Prokaryotes evolved first, and then eukaryotes. Culture. So we often use culture colloquially to mean, like, your system of traditions, but in microbiology, culture means to grow bacteria, or like a petri dish full of bacteria is a culture. And the jello in the petri dish, we call it the culture medium, the stuff that the bacteria grows on. So to culture bacteria is to grow bacteria. It can be a verb or a noun. You can culture a bacteria culture. Inoculation means to add bacteria to a culture. So if you take a swab and then you swab it on a petri dish, you have inoculated that culture medium. You've inoculated that agar. You also hear the term inoculation when people get vaccines, because when you're getting a vaccine, you are introducing a live or a weakened or dead bacteria or virus. So you are adding microbes to a somewhat sterile system. So it's also called inoculation. Enriched medium, again, medium is just the stuff that we grow bacteria on. It can be solid, like a jello, or it can be liquid, like a broth. And a lot of times it's pretty simple stuff, like basically like chicken broth. But sometimes we have bacteria that need more nutrients. They're extra picky, so we need to use enriched medium. So enriched medium just has extra good stuff added to it, like blood or milk. Fastidious is a word that kind of means picky, I suppose, and stubborn. Stubbornly picky, maybe. So bacteria that are fastidious are stubbornly picky about where they will grow, what nutrients they need. They require enriched media. They won't grow on some simple chicken broth style media. Selective media is cool because it helps you in the investigation process when you're trying to figure out what bacteria you have. Selective media, it selects for only certain microbes to grow. So it'll have ingredients that inhibit growth of everything else. So only certain types of microbes can grow. So it kind of helps to clear that background noise of other stuff you're not interested in. So a lot of times antibiotics can be a selective media. So things that can't, or salt is really common in selective media. Only certain bacteria can withstand high salt concentrations. So adding salt will kill a lot and restrict the growth to ones that can tolerate, and select for ones that are salt sensitive or salt resistant. Differential media makes the things that grow on the plate look different, so you can distinguish them with your eyes. Usually they involve some kind of dye or pH indicator, which is like a dye that changes colors with different pH, so that bacteria maybe that produce an acid will grow one color and bacteria that don't grow another color. So differential media still grows most things, but it helps you distinguish them visually. A colony is a growth of bacteria on a solid medium, like an agar plate. So it looks like just like a little bump, usually kind of a glossy little dot. It originates from one single bacteria that landed on that spot, just kept growing and growing and growing, and all the generations just landed on top of it until you had enough that you could actually see with your eye. So a colony is actually, it originates from one bacteria, but a colony actually represents millions of bacteria. So, resolving power, now we're getting into microscopy. Resolving power is the ability of a microscope to distinguish two different things. So if you have two things that are close together, it's about resolution. So you may know from like working a camera that if something's kind of blurry, you can't see it clearly, then it's low resolution. So you don't want your microscope to be low resolution. You want it to have a high resolving power, an ability to see things clearly, not fuzzily, to distinguish two things that are close together. When we stain bacteria that are on a slide so that we can see them better in a microscope, there's different types of stains. So there are positive stains, and positive stains actually stain the bacteria. Bacterial cell walls, because they're made up of phospholipids, which are negatively charged. So the cell membrane is made of phospholipids, is negatively charged. So positive stains stick to the membrane because opposites attract. So the positive stains stick to the bacteria and stain the bacteria so you can see them, so they look blue or pink or whatever color, green. A negative stain is negatively charged, and it's repulsed by the negative charge of the membrane. And so with a negative stain, it doesn't stick to the bacteria, it sticks to sort of the surroundings. And so you get sort of this, clear area. The bacteria, it's kind of like photo negatives. When you take a photo and you look at the negatives, the light areas are black and the black areas are white. So the bacteria in a negative stain appear clear. They're the only thing that is not stained. And then in a differential stain, this uses two different colors of stain to differentiate between two different types of bacteria. So a common differential stain that we use is the gram stain method. There's a blue stain and a red stain. You add them both, and basically whichever one sticks determines whether it's gram positive or gram negative. It's more complicated than that, but that's an example of a differential stain. Alrighty, that's enough. The end.